So what a thrill to be here as I look out, and what a thrill to be back in Dallas and in this really beautiful George Bush Library. And it's a particular thrill to be able to introduce you to our panelists tonight, two of the most renowned researchers and people in the field of neuroscience. And let me start off with Roger Rosenberg. It's really a thrill to be having Roger on my side here. Roger is the founder of Department of Neurology in 1973, and he's been working at it for 45 years since 1973. It's really unbelievable, and continues to dedicate his efforts, his intellect, his influence to do what he can to have influence on dementia, and in particular on Alzheimer's disease. But that's not where it all started. He made major contributions in the 70s when he was the first to describe a genetic disorder that presented with, again, instability, progression, for which today, again, there is no obvious cure. Dr. Rosenberg is esteemed. He's trained over 200 neurology residents in this city, and he's a legend. I've known Roger for a long time, looked up to him, had numerous manuscripts rejected by him when I've submitted to numerous journals. But we've still remained good colleagues and good friends.
He took all mine.
He took yours.
Yeah.
Rudy says he took his, never took mine. But it's a thrill to have Roger as part of this in his hometown. And then, of course, it's a thrill to again introduce to this audience Rudy Tanzi. Rudy's a legend. Rudy, you know, when you hear what he's done this year, so he's the author of this particular book that just came out on the healing self with Deepak Chopra. He's played keyboards this year with Aerosmith, been in all kinds of major concerts. They think I'm joking, but I'm actually not.
It was a strange year.
And including with the South African wonderful guitarist, Johnny Depp. But for tonight, Rudy and I knew each other in the early 80s when I moved from South Africa to Boston, Harvard Medical School, and I met this wild, wiry, incredibly creative, and amazing scientist, Rudy Tanzi. And Rudy went on to clone 3 genes for Alzheimer's disease. He's made major contributions all of the way as we understand the target, the biology of Alzheimer's disease, how it works, which are the targets. And so we're really thrilled to have these kind of people participating in this important symposium, Roger and Rudy. And let's welcome them both. So tonight, we're actually talking about something very serious, something that affects perhaps up to 500,000 Texans now, 5 million Americans, and close to 50 million people around the world. A disease which robs you of who you are, robs you of all that you were, and creates despair and darkness in the families and in the society. The good news for us is that many of us will now live till our 80s, but the bad news is that 1 of 3 of us won't know we got there. We won't know this, and all of us will know somebody who's suffering from Alzheimer's disease, somebody who's unable to communicate, unable to recognize the dearest people that are close to them. And maybe just as a start, I'd like to read you just a paragraph from Lisa Genova, somebody else who's been in these forums before, from her New York Times bestseller and also was awarded the Academy Award, Still Alice, where she describes just what we're talking about, which we all know. And what she says, with talking as a Harvard professor now, talking to an audience, she says, "My reality is completely different from what it was not long ago. It's distorted. I struggle to find the words I want to say and often hear myself saying the wrong ones. I'm losing my yesterdays. If you ask me what I did yesterday, what happened, what I saw and felt and heard, I'd be hard-pressed to give you details. And I have no control over which yesterdays I keep and which ones get deleted. But she goes on then to say, I still have hope for a cure for me, for my friends with dementia, for my daughter who carries the same gene. Please don't look at our scarlet A's that are inscribed on us and write us off. Look us in the eye, talk directly to us, never give up on us. And that's what this is about tonight. We are not going to give up on the tsunami of dementia that's affecting so many people we know and will affect so many of us as we get older. And so tonight is just a step in the direction to overcome this and to do what we can together to promote research, to do what we can to overcome what is a black cloud that threatens to smother so many of us. So let's start. Let's start with, Roger, I'd like to ask you first. Maybe just as a baseline, tell us, what is Alzheimer's disease?
Thank you very much, Michael. I just briefly want to express our collective thanks to Leslie Crozier, to Catherine Wynn, to Carol Hall. Great privilege for me to be here with Michael Hayden and Rudy Tanzi, and thank you, ladies and gentlemen. So, Alzheimer's disease is a neurodegenerative disorder. It has strong genetic underpinnings to cause it. It begins in many different ways because the genes that produce it have different variations, and they can express themselves clinically in somewhat different ways. But there seems to be a common denominator in which there's an impairment in memory ability, amnestic memory ability, to recall events, names, peoples, circumstances. Memory leads the way in most individuals, but it doesn't stop there. We all forget something, where we put an object or forget the name of a person. But Alzheimer's is progressive. It continues to be unrelenting progression and loss of memory, and then executive functioning, frontal lobe functioning in terms of reasoning, judgment, insight, abstraction, and understanding. These are very important characteristics that we just heard Dr. Hayden describe in that patient. It also involves language functions. Individuals have difficulty finding the word as they try to express themselves. They begin to speak and then they stop. They can't find that word, and it takes time to find the next word. It goes on to an impairment in grammar, in syntax, making a paragraph of reasoning and thinking. And it goes on to become, at some point, even mutism in terms of your ability to express yourself and understand. It can also affect non-language and non-verbal and non-executive aspects, and that is your spatial orientation, where you are in space. Lived in the same house for many years, but where is the living room in relation to the next room? You lose, if you will, the map of the world. And it is progressive. And it involves not only genetics, but hopefully some of these treatable biomarker events, that is hypertension, diabetes, lack of exercise, obesity, smoking, and attention to a good education and a good nutrition that can be treated that might decrease the rate of progression. But underneath it all are a variety of genes, perhaps 50 genes, and perhaps more to be discovered, that have variations in them that give the different presentations that I've alluded to. So keep in mind, it is a neuroprogressive degenerative disorder that involves verbal and nonverbal aspects of the brain, resulting in shrinkage and atrophy of the brain, preceded by loss of connectivity and circuitry, loss of connectomes that mediate information storage and retrieval.
Thanks, Roger. So, Rudy, we've heard about more the clinical sequelae of having Alzheimer's, but you've been an expert in defining the pathways involved, from plaques, tangles, and then neuroinflammation. Give us a perspective now on what you think causes Alzheimer's disease at the cellular and molecular level.
Well, first of all, I'd also like to thank all of you for being here and supporting Alzheimer's research and for inviting me from the smallest state to the largest state on the mainland. I originally came from Rhode Island. I grew up. I've been in Boston my whole life. But picking up where Roger left off, let's think about how the brain works. How does memory actually get stored? We take for granted that sensory signals come in. You're hearing things, seeing things, smelling things, touching things. But think about an infant baby who sees red shoes. There's no context, right? The baby sees red shoes and doesn't think, oh, those go on your feet, someone's going to wear them, they're red, that means either they're women's shoes or they're Deepak Chopra's shoes, because Deepak always wears red shoes. But the baby doesn't know all that, right? All that information is learned. All your life, as you have experiences, you're making associations and learning what goes with what. You know that your keys go into a certain dish when you come into the foyer or into a drawer. You don't put the keys in the refrigerator. We take these things for granted, that there's context as sensory information comes in and is turned into information. Sensory signals get turned into information. In Alzheimer's disease, that's happening. The sensory informations come in. They get stored here in the hippocampus, which means seahorse in Greek, because it looks like a little seahorse. But then when you want to recall them, because you can't put them in context with what you already know, that's what learning is. That's why we say it's a disease of learning and memory. It's forgetting, and it's also not having recorded the information correctly and putting it in context. That's why you might find the keys in the refrigerator. I think there's a scene like that in Still Alice.
Yes.
So why is that happening? Well, the first thing that happens in the hippocampus is that it starts to gradually get disconnected from another part of the brain that's behind your nose, the entorhinal cortex. That's kind of the way station where sensory signals come in. And picture a big cable. It's called the perforant pathway because it actually take that seahorse and then spear it, perforate it. And that's how the sensory information comes in. And that's where you start to get the pathology. The first pathology is plaques, amyloid plaques, which are these big, sticky, boulder-like material outside the nerve cells. And back in the day, not long after I met Michael for the first time in Jim Casella's lab when we found the Huntington's gene back then, very exciting days. It was the first time a disease gene was found. And I remember telling people in the lab, Michael Hayden, he literally wrote the book on Huntington's because there was a book on Huntington's and it was the only one, and Michael wrote it. The blue book, I remember it was blue. I remember that. And soon after that, a clone discovered this gene I named amyloid precursor protein. Not a very imaginative name, but it's the protein, the gene that makes the protein that gets clipped up to make the amyloid plaques. And soon enough, by the 80s and 90s, there were 4 different genes for Alzheimer's. 3 of them caused the early onset form that strikes under 60, familial. Those mutations, there's about 300 or so of them. They guarantee the disease. And then there's another gene, ApoE, for the late onset. But what's important is that all 4 of them led to the same pathology, plaques. In different ways, it was excess plaques in the brain. And how many of you who follow Alzheimer's have heard about the controversy about whether plaques cause the disease or not? Raise your hand. Yeah, so that's how, this has gone out of science into the popular media. Well, it turns out that the big reason for that controversy was that we put these genes into mice. The mice got plaques, but they didn't get the other pathology that actually kills the nerve cells, the tangles. The tangles are these twisted fibrils that form within the nerve cell. The nerve cells didn't get tangles in the mice. So if that didn't happen, we said, how could the amyloid be causing the disease? Well, fast forward now to a few years ago, and if you take human stem cells, turn those into nerve cells, grow them in a mini brain, organoid in a petri dish like we did in my lab, and now you can test, does amyloid cause the tangles in human nerve cells in the facsimile of a mini human brain organoid? Yes, they do. The problem was, it was mice, right? Mice are not people. Newsflash for biologists. We take mice too literally. They're mice, folks. So now that we get these brain organs, we can see plaques cause the tangles. But then we learn something else, and I'm getting to the end. And that is that once in a while you get these resilient brains. These are people who die, they're perfectly fine, they happen to have an autopsy, and the pathologist sees tons of plaques and tangles and say, this must be an Alzheimer's case. Nope, they were perfectly fine when they died. And what's always missing in these very rare resilient cases is the third pillar of the pathology, neuroinflammation. This is where the brain's immune system is recognizing these plaques, these tangles, these little brush fires of cell death, and react against it, trying to wipe out that area of the brain. It's been compromised. That process of neuroinflammation, the brain's own primitive innate immune system doing this, kills 10 to 100 times more nerve cells than the original plaques and tangles, and that's when you go down the slippery slope. So the good news is, from resilient brain studies, is you can live with lots of plaques and tangles, but when neuroinflammation kicks in, that's when you go down the slippery slope. So a lot of our work now is focusing us trying to stop the neuroinflammation, because that's how you can help patients right now. If we want to stop this disease 20 years before symptoms, you hit the plaques and the tangles. And a lot of these trials that fail were because we didn't hit them soon enough, but we'll probably get into that.
Yeah. Okay, so thank you. I think that gives us a good background about both the pathways involved as well as the clinical symptoms. The question is, how do we make the diagnosis? What technology can we use? And are there ways to know when somebody truly has Alzheimer's besides the forgetfulness? You know, pretty much everybody in this room has sometimes forgotten words, and it doesn't mean you have Alzheimer's disease. So how could you diagnostically make the diagnosis more certain that you have Alzheimer's disease? What would you do? What technology would you use?
Thank you. The occurrence of memory loss is not specific to Alzheimer's disease. The occurrence of dementia is not specific to Alzheimer's disease. There are a number of entities that produce these kinds of symptoms non-specifically. Frontotemporal dementia, Lewy body dementia, plaques and tangles for Alzheimer's, yes, TDP-43, strokes. But Alzheimer's disease can be diagnosed more specifically. As I indicated, it is a progressive disorder. It involves multiple aspects of behavior, as I've outlined. Patients come in and give you this history that it's a regular progression. You do a careful neurological exam to make sure that they don't have focal findings that would be suggestive of a stroke or a history of something suddenly occurring. Vascular dementia does occur. You want to get an MRI. You want to see that there is a diffuse impairment of the brain involving the structures that Dr. Tanzi referred to. Shrinkage and loss of the hippocampus, the mesial temporal lobe, where amyloid and tau begin to occur in the entorhinal cortex. You want to see atrophy also in the parietal area and the left side, where language is important, and then the non-dominant side in terms of spatial orientation. So these characteristics are important in terms of the neurological history, the symptoms I've referred to, and the absence of things like strokes and hypothyroidism and so forth. But the specificity is to try to determine if there is the accumulation of the factors that Dr. Tanzi referred to, amyloid and tau. So you can measure the cerebral spinal fluid, and you can measure the levels of which A beta 42, which he referred to, which is the clipped out portion, the short portion, 42 amino acids of the long amyloid precursor protein. And you see in individuals with Alzheimer's disease, as the symptoms progress, the levels of A beta 42 fall. They go down because the deposition of amyloid is occurring in the brain and sparing the cerebral spinal fluid, or the water that you can measure with a spinal tap. So that's first thing you can do is measure A beta 42 and the ratio of A beta 42 to A beta 40, which is another species of amyloid which is clipped out of amyloid precursor protein. The second measure for specificity would be to show that the level of phosphotau in the cerebral spinal fluid is elevated. And there is a formula of decreased A beta 42, the ratio of A beta 42 to 40, and the elevation of p-tau in the cerebral spinal fluid, which gives you a high degree of specificity for Alzheimer's disease. These manifestations occur 25 years before clinical symptoms, specifically the reduction of A beta 42, and Dr. Tanzi referred to. This is a long preclinical state of the disorder before symptoms begin, and it's an important window of opportunity to develop therapy before there's neuronal loss, before there's dementia. That's the time you want to act and find a specific therapy for amyloid and also a specific therapy for tau. And the accumulation of tau occurs after the accumulation of amyloid, as you heard. And amyloid and tau are important. There's not one or the other, as he said. Amyloid diffuses through the brain early in the first 10 years of the disorder, asymptomatic individuals. And it is permissive for the subsequent diffusivity of tau. If there isn't amyloid, you don't get that diffusivity of tau, the spread of tau, and the correlation with dementia. Dementia correlates, as he said, with the rise of tau, but the two are together, and one precedes the other, and both is necessary. The next thing you can do is 10 years after symptoms occur, you can do an amyloid and a tau imaging of the brain. You can demonstrate that with the radioactive 18-fluorodeoxy form of a marker that binds to A-beta. You can show non-invasively that amyloid is diffusing through the brain from the frontal lobe to the parietal lobe and from the temporal lobe. And then subsequently, tau diffuses through the brain. So to answer the question, you've got to have a baseline that is compatible with Alzheimer's disease, that you've ruled out other neurological disorders, and the specificity is the measuring of A-beta amyloid, the measuring of tau in the cerebral spinal fluid, and then years later, after that begins, to demonstrate the specificity of amyloid and tau diffusing through the brain. Non-specifically, you can do a glucose or sugar scan in the same way and show that metabolism is impaired and it is decreased. So it is the amyloid and the tau that gives specificity, both in terms of examination of cerebral spinal fluid and brain imaging.
So the bottom line is there's a way to actually even know before signs and symptoms become dominant that you might have Alzheimer's disease. And this can be seen quite early as one looks and starts thinking about these things. Now, Rudy, you know, it's been a tough road on the therapy side. I mean, and today again, as you just pointed out to me in the New England Journal, more discussion on certain approaches to treating Alzheimer's disease. And for people with families who have or know of friends, this could be a depressing time. Let me ask you, why have these trials, if these are the mechanisms that you've defined, and the trials have been directed towards these mechanisms, why haven't they worked?
Well, you and Roger both hit on it. One of the things that the elephant in the room is we don't diagnose Alzheimer's disease until there are symptoms. Imagine if we did that with cancer. Imagine if we did that with heart disease or diabetes. Right? In neuropsych disease, we wait for symptoms. We wait for the organ to degenerate to the point that it doesn't work right. We don't do that with other diseases. So things have to change. We need to start detecting the amyloid by imaging or the spinal fluid 20 years before symptoms. I say, if you're from a family with Alzheimer's, take the youngest age of the first-degree relative with the disease, subtract 25 years, and that's when you'd first want to have your amyloid scan. Maybe in the future, once we have a way to handle amyloid safely, by 50 years old, it's like a colonoscopy or a breast exam. You just have it. At this point, you'd have to have your brain imaged to see if there's too much plaque. The big bottleneck has been, and the reason why these trials on amyloid fail is very simple. I like to think of it this way. Amyloid is the match. The tangles made of tau are like the brush fires that spread. Then neuroinflammation, inflammation is the forest fire. With these amyloid trials, we were trying to put out the fire by blowing out the match. Now we know that. But then you'd say, okay, so we got to do these trials earlier. But what company is going to do a 20-year prevention trial? Think about that. It would cost $10 billion, and by the time it's done, you could say, at best, congratulations, your drug's now a generic because your patent life is over. I mean, it doesn't work. And the good news this year, which we could talk about, is the FDA, under Gottlieb, announced in February new guidelines that for the first time will allow us to start doing trials in people who are symptom negative, pathology positive. We say there are 5 million patients in this country.
I'm going to come back to the regulatory environment.
Okay, okay, right.
Is a very important, opportunity with changes in terms of regulation. But let's stay with...
Let me say one last thing. 5 million patients with symptoms. How many people in this country have pathology on their way to symptoms? Probably 25 million. It's probably the most common disease in the country, if you judge it by cancer or diabetes terms.
So what Rudy is saying is that if you want to treat heart disease, measure cholesterol first and lower your cholesterol before you have the heart attack. And in the same way here, he's saying, let's find a way to measure early symptoms, early signs, and get Alzheimer's disease before it starts. Now, the challenge is really how we'll do that as a society. How will we really place dementia, place Alzheimer's disease at the highest priority so that we actually have the appropriate investment? Because I think Rudy's right. For drug companies, this will be a very hard, difficult program to buy into when you have to treat for so long before you see a result. And so we have to look at other ways to define progress. And let's just talk about the FDA for a moment now, because under Scott Gottlieb, we've seen tremendous progress in treatment of cancer and approval, early approval. And the new guidance that just came out a few months ago has offered new approaches that are going to be treating not only based on diagnosis, but on surrogate endpoints, which include some of the markers that you've heard about. So let me ask you really what your view is on the FDA guidance and what gives you hope with that in terms of clinical development now, Rudy?
Yeah, if you read the wording of it from Billy Dunn, who's the head of neurology products, it says that for research purposes, you wouldn't want to do this for medical diagnosis purposes, but for research purposes, you can do a trial in what you would call stage 1 Alzheimer's based on someone who has the pathology but doesn't have the symptoms. I mean, think about cancer, right? When someone has a neoplastic cell in a small tumor with no symptoms, we say we're treating cancer. We need to do the same for Alzheimer's. So they'll let us do those trials, and this language says if we can move, for example, amyloid. Let's say you have people imaged for amyloid plaques, they're symptom-free, and you show a drug safely moves the amyloid back down, they will consider approval on that trial. You don't have to do a 20-year trial to say, let's make sure they don't get Alzheimer's later on. And then once the drug is out, and I read these guidelines about 10 times to make sure that what I'm saying is correct, that once the drug is then approved, post-approval, they'll keep track and say for some period of time, 5, 10 years, now let's see, now let's do the test in the public with a safe drug. Let them take the non-drug to stop amyloid, and let's see over the next 5 or 10 years if we drop the conversion to symptoms in those people. But let's not make them wait 20 years for a trial that nobody else would do. So I say my hat's off to Gottlieb and Dunn for allowing us to do this, and I think it's a new era in Alzheimer's research because of this. So I'm very, very optimistic about it.
I would agree.
So that makes me not as depressed anymore.
So we
Probably could tell.
really doesn't look depressed to me. So we've had some questions coming in from some of you, and I'm going to ask Jimmy Westcott and Steve Crozier to come to the microphone. These are two related questions.
Is there a microphone?
Or can we give a microphone?
I can speak from here, I think.
Okay.
We'll repeat the question.
Okay, Jimmy, so your question. Do you want to ask your question?
Yes, my question involves the percentage of the population. Does a country like America or India, do we have more Alzheimer's patients already recognized than other countries?
So maybe, Roger, this is a good one for you. The question, the incidence of Alzheimer's, how does it vary around the world, and what determines the different prevalence, if there is one?
It's a complex question. Is Alzheimer's disease uniform in all populations? What is the epidemiology of Alzheimer's disease? Is it uniform or not, and what would be the reason? It is not uniform. There are major differences around the world. Some important epidemiological studies have been published that I've had a chance to look at. And if you look at the burden overall in the world of Alzheimer's disease, it is significantly increased, 3- or 4-fold in Asia, in China, in India, and in Southeast Asia. Not because the populations are larger, but the percent of individuals per 1,000 in the population is also increased. The evidence of the statistics that I've referred to for these countries is reproduced in a number of studies. It does not seem to be genetically driven, the polymorphisms, the variations in the 50 or so genes I've referred to, because Chinese Americans and Indian Americans from India and Cambodian and Vietnamese Americans don't have a threefold increase of Alzheimer's disease. It is related, and the statistics correlate with the modifiable, treatable disorders that I have referred to. Cerebrovascular disease, silent strokes, not so silent strokes, the occurrence of uncontrolled hypertension, uncontrolled and poorly treated diabetes, elevated cholesterol and lipids, smoking, lack of exercise, obesity, and even poverty in these countries, in rural areas, resulting in impaired education. The anatomical reserve in the brain is extremely important. The brain needs to form all of the proper connections for its potential and to be able to perform normal behaviors. This anatomical reserve is protection against Alzheimer's disease. You find individuals who are highly educated seem to be protective against those who don't have that luxury. So education in the third world countries I referred to, very important. In fact, Sunday in the New York Times, there was an article on the front page of children in India having major difficulty with their education because they can't get glasses. Simple things like that. Access to medical care, access to the treatment of these disorders that add to dementia and increase the rate of Alzheimer's disease. And the other is diet, malnutrition. Malnutrition, protein, in terms of adequate protein in the developing brain, in utero, as well as in the first years of life. Now, China and India are improving. That's the good news in terms of Deng Xiaoping and Xi Jinping currently increasing the economics and access to good care in the rural areas, in Modi in India. Europe has the next highest rate, particularly Eastern Europe, for the same treatable disorders that I've referred to, then Europe, Western Europe, then South America, a bit less, North America. And Africa is listed lowest. But these figures are thought to be artifactual because there's just not adequate reporting. Attribution bias. Patients are just not being seen and cared for. And countries are trying very hard in order to provide care for patients in sub-Saharan Africa. So to answer your question, the distribution is very unequal, and it seems to correlate best with these treatable disorders, modifiable disorders that increase your risk for Alzheimer's disease.
So,
Can I just say that that was the best answer I've heard to that question in my life? Because I don't know how to answer it, so that's what I say. That's amazing.
Steve, is Steve around? Steve Crozier?
I'm here, yes.
Okay, Steve, your question.
Brother.
And it's actually for Roger too, I think.
Yeah, well, and Dr. Rosenberg diagnosed our mother years ago with Alzheimer's, and so just to pre-qualify this question, I've never been president of anything, so that's yeah, that's Leslie. So, but my question is, once one has been clinically diagnosed with Alzheimer's, what are the time frames moving forward? In other words, I know there's early onset. I know you diagnosed my mom when she was in her mid-80s, so that's not early onset. But, you know, there's, as you know, this affects the families and everybody around. Is there time frames for the progression or the disease as it gets worse and worse? Is there any quantifiable data or thoughts on the future once one is diagnosed?
And how does it vary between people?
There's a great deal of variation, even within the same family, and even in terms of siblings. There's great genetic diversity between each of us, and genetic diversity between siblings. We have a wide variation in gene expression in addition to our diversity. I referred to the variations in a subset of genes, and these variations increase your risk for Alzheimer's disease. Not one alone or a dozen alone, but it may require 50 or hundreds, maybe even thousands of the 21,000 genes we have in our genome. And they determine the phenotype. They determine the age of onset. They determine the rate of progression that you've alluded to. And they determine the phenotype or the variation in the clinical manifestations of the disorder. The genetics are extremely important, these variations that determine the kind of Alzheimer's disease a person will have. That's one set of genes that are very important. And the other is the environment in which we live, in terms of our experience, in terms of siblings and what they experience, in terms of infections they will have, or other diseases they have, or hypertension, or diabetes, or obesity, or lack of exercise. These are important factors in the environment that are encoded in our genome, a specific set of genes referred to as the epigenome that are interposed between the environment and translate what is happening to you and change the expression of the rest of your genome to respond to that. And so your genome is very different in terms of its expression. It's also different in terms of the diversity of the genes you inherited. It's both nature and nurtured, if you will, to quote Francis Galton, who used that term for the first time, a cousin of Charles Darwin. So there is important differences in terms of gene expression that you inherit and important differences in gene expression that you experience in life. And we think that's the reason for the variations in the diversity of the clinical disease itself.
So Rudy, Roger's talking about resilience and how the brain can be more resilient and what we can do. There's genetics, but there's also things in our environment that we can modify, even now, each one of us, to modify the presentation and potentially enhance the reserve of our brains. And you've written about this. You've spoken about the callous and relentless thief of identity and thief of life and mind and self. So for people here today, what could they start doing themselves, even now, to build resilience of the brain, to do something to try and, if they're destined to get Alzheimer's, what could they do to delay it, to have more defense against the onset of Alzheimer's disease?
Well, you know, probably about 8 years ago, when I met Deepak Chopra, we spoke together at a TED conference. And it was a weird way to meet him. We were in the men's room, and he was next to me, you know, doing that thing. And he turns to me and says,
I don't think we want to ask what thing.
Yeah. he said, I really liked your talk. And he said, tell me, is the brain a noun or a verb? And I said, can we talk about this outside? But anyway, I decided, you know, over time that this would be a great way to write books. I mean, while we're waiting to get these drugs and get everything together and get our act together to actually treat this disease, prevent this disease, I thought it'd be a great time to write some books about it. So we wrote Super Brain, Super Genes, and now The Healing Self. And in The Healing Self, I'm not trying to sell the book, I'm just answering the question, by the way. At the end, we have what's called a 7-day plan, where each day you focus on one thing to help protect your brain. And in my opinion, the main enemy is neuroinflammation, because plaques get you there, tangles get you there, but the resilience studies say that neuroinflammation is what takes you out. So if you look at what affects inflammation in the brain, and you want to be able to stop it, in terms of lifestyle, I use an acronym called SHIELD, shield your brain, where the S stands for sleep. Sleep 8 hours, at least 7 to 8 hours. And it doesn't have to be continuous. You can, as long as, right after REM, after dream sleep, you go into this deep, slow wave sleep where your brain's immune system, the same that can turn against it, can be housekeeping and clean up the brain. So in that deep sleep, I like to call it mental floss. Your brain's cleaning itself. You got to allow enough dream, you know, REM, slow wave, deep sleep cycles, even if it means taking a nap where you dream and have a slow wave sleep during the day if you don't get enough sleep at night. H is handle stress. We found, you know, we did a trial of meditation on just gene expression, like you were saying. We looked at the expression of all 22,000 genes in people who were meditating intensively for a week. And I wouldn't have written these results in a science fiction book. The inflammation genes are down, the genes that control amyloid deposition going in the right direction. Telomerase, the anti-aging enzyme that controls the tips of chromosomes, Elizabeth Blackburn won the Nobel Prize for. Telomerase activity was up 20 to 40% after a week of intensive meditation. So handling stress, because stress is a killer. And then I is interact with others, staying social. Loneliness is a risk factor for Alzheimer's, not being alone, but being stressed out about being alone, loneliness. Exercise, probably nothing better than exercise, moving. Exercise helps induce BDNF in the brain. It helps reduce inflammation. It helps induce enzymes that get rid of amyloid. Just about everything you want good to happen in your brain comes from exercise. And it doesn't have to be intense. It can be, a brisk walk for a half hour to an hour a day. Or make sure you stand up at least once every hour, so the time's going to be coming up soon to stand up. L is what you're doing right now. We're actually protecting you from Alzheimer's because you're learning new things. Unless you're sleeping and you're still being protected, so that's okay too. And so when you learn new things, you make new synapses, right? So I say when you're going to retire, think about financial reserve and synaptic reserve. The more synapses you have, the more you can withstand getting dementia. So synapses, when you learn new things, you make new synapses, you strengthen old ones, and what really correlates, Roger will tell you, with degree of dementia is loss of synapses. And finally, D, diet. Mediterranean diet, over and over, Mediterranean diet is the most protective. Take care of your gut microbiome, probiotics, yogurt, or a probiotic supplement. The gut microbiome controls inflammation in the brain, and we're learning more and more about the gut-brain axis every day. So that's in a nutshell.
Yeah. I think that's very, very helpful for each one of us to think about how we can take steps every day to prevent and just prolong healthy lives, and in that way also prevent dementia. Now, we've spoken about individuals, but society, we live within communities, we live within societies, we live within states, we live within countries. And the real question I have is how do we also help to facilitate a better, fairer, safer world for people with dementia in communities? And this means, and I just wonder about your thoughts about the stigma. What do we do to reduce stigma? How do we create a fairer world for people with this disability? We're doing quite well as a society with people with physical disabilities. But with mental disabilities, we still, it's lagging behind. And communities, and so I'm wondering about your thoughts about what we can do as communities to create safer, fairer worlds for people with dementia, because we need to have a society that's more compassionate in that way and reduce the stigma. And I wonder if you want to talk about that.
Well, I think we should state our case. I think we should make Alzheimer's disease evident to friends and family. It shouldn't be something we're ashamed of. Cancer has that stigma in the past, and I think they've overcome it, and they're very articulate with the Congress to get adequate funding. We're just beginning to state our case. Alzheimer's funding at the NIH is beginning to increase, finally, $1.8 billion. That's not the $6 billion for cancer. So I think each and every one of us need to express positively that, yes, my loved one, my friend, myself, have Alzheimer's disease, and I want to get your support. I want you to understand what I'm going through. The quotation that we heard, the human side of what we are dealing with and the suffering and the bleakness of the future. We need to be encouraged. President Reagan was, I think, a role model in that regard to bring it out of the depths and to allow it to be expressed. And so I think mutual support by friends and family, very important. Very important that we get funding responsible for Alzheimer's disease. Dignified changes in the household to make it convenient for the Alzheimer patient, to protect them. They don't fall, they don't get lost, that they're supervised, that this is something that we need to help. We need to support good insurance across the country. As a democracy, we need to do better to provide this kind of long-term care as part of a right of citizenship and not just a privilege. So I think these are some steps that could be taken that would make a difference to make life better for patients with dementia.
The interesting thought for all of us here is within each of our communities. So how would we make Dallas more dementia friendly? Just as a rhetorical question for now, but as a community, how would we want the banks to help, the libraries? How could we keep people functional for longer whilst we demonstrate the compassion that we all feel for people with this illness? Rudy?
Yeah, I just want to comment, Saturday night, I was at an event in Rhode Island to have this Rhode Island Hall of Fame, and I was dreading it. Patrick Kennedy and I were inducted into it on Saturday night. It was great. My mom was there and everything, wore a medal. But, you know, as it turned out, it was a really cool event because Patrick Henry and I were talking about this idea of stigma of mental illness, stigma of addiction, stigma of neurological disease. And what we talked about was the idea that from childhood, and the way Patrick put it was very interesting because, you know, Patrick suffered himself from addiction and issues that he overcame. And he talked about having every year from childhood, as early as possible, a checkup from the neck up. So he called it a checkup from the neck up. The idea that you need to keep your brain healthy because what we used to call a neurochemical imbalance, well, it starts with pathology like inflammation. It's pathology. When there's an extremist act from a radical, you can bet there's a lot of inflammation in that brain. Inflammation drives depression, it drives autism, it drives Alzheimer's, ALS, Parkinson's. So if we had some way in the future to just say, you know what, I got to keep my brain healthy so I can stay mentally healthy, and this becomes a way of life from childhood, and you start checking early on whether there's some pathology in the brain and nip it in the bud early on, it no longer becomes a stigma like there's something wrong with you. It becomes, oh, it was like some pathology that built up, and you know, we hope we can help it. But I think we have to move in that direction of pathology is not stigma, and it's not being, mental illness has to do with pathology, not how smart you are or how calm or excited you are. You know, I think we have to make that jump.
This is a provocative concept, which is creating societies that foster healthy minds and healthy brains.
Yeah.
Both. And
recognizing
actually,
the pathology.
just as we have gyms for improving exercise, which does contribution, but there's more that we're asking for healthy minds and healthy brains. It's not just diet. It's
and
not
keeping
just
track
exercise.
of it.
Keeping track of it, and also being able, just as we're talking about cardiac safety and cardiac prevention, to have a society that's really fostered on fostering healthy minds and healthy brains. And this is just it's a shift. We're doing small pieces of it as a society, but not completely.
When we were growing up, there was a C word, right? Cancer was the C word. People didn't say cancer, right? Now it's not a stigma, it's pathology. And we got to get to the same spot with Alzheimer's and mental illness.
So let me ask Stan Levinson. Is Stan here? Because you had an interesting question about, and just I'll introduce that question just to say, of course, we talk about Alzheimer's disease, but you know, in 2010, when David Cameron was Prime Minister of England, and there was a big meeting in the UK with the G7, they came up with a contract about dementia. It actually wasn't about Alzheimer's, it was about dementia, talking about having some way to create societies that were friendlier to dementia by 2020, and then having potential drugs that would influence dementia, truly modify the course of the illness by 2025. So Stan.
Well, first let me thank all of you for such an informative and helpful and inspiring discussion this evening. Just very grateful to you. More on distinguishing Alzheimer's from dementia. I think you've explained it medically very, very well, but we're wondering if the rate of suffering from Alzheimer's equates to the same rate as one with dementia. And if so, the progress of getting well and knowing ultimately what the outcome is going to be. But if you could share with us some more perspectives on those related issues, we'd appreciate it.
Well, dementia is a symptom. It refers to what I indicated previously: memory, executive function, reasoning, abstraction, language, spatial orientation. It's not a disease. Alzheimer's is a specific neuropathological and clinical disease. So it is one of the dementias, and it is the leading dementia cause of dementia in the United States. Approximately 70% of patients who have dementia, the symptoms I've referred to, have Alzheimer's disease. So we want to make that distinction. There are other causes of dementia that I have referred to. Now, the variation in terms of the age of onset, the rate of progression, the kinds of symptoms, what a patient can expect is very hard to predict because of the various genetic aspects I've referred to, the various environmental circumstances a person faces, the other medical comorbidities a person has, the support that a person has in the family and friends in terms of sustaining them, of providing them with therapy for these other medical disorders that add as a burden to neurological disease and a burden to the brain. Cerebrovascular disease in particular, you want to reduce that as a burden. So it's extremely important to try to reduce the rate of the disease if you can by treating these comorbidities, including depression, providing a positive, supportive atmosphere. That makes a critical difference. But it is true. Patients ask early on in the evaluation that I and others provide, is this Alzheimer's disease? Is this dementia? Are you sure that it is? Is it something treatable like stroke? And what will be the rate of progression of this? What's going to happen to me? What do I have to plan for in the future? And we try our very best to reassure and offer hope. That's extremely important. But in actuality, it's very hard to determine the rate. If someone has had symptoms for 10 years and we see how they're doing after 10 years, then you do have a correlation of the rate of progression that they have had. That's no guarantee that that slow rate of progression will continue, or that it will speed up or slow down even. But we try our very best to predict what will happen based on what has happened before. And the variables of genetics and nature and nurturing in the environment are very hard to predict. We do the best we can, but you are correct. We have to offer hope, and we do.
So let me just add a bit to that and then ask Rudy his thoughts. You know, when we look at the pathways involved in different forms of dementia, and Parkinson's has dementia, Huntington's disease has dementia, Alzheimer's, of course, and even pathways with ALS or Lou Gehrig's disease, what you see is there's often convergence of pathways. And so sometimes you can look at rarer diseases that give you insights into pathways that then allow extrapolation to more common disease. And sitting in Dallas, we have to acknowledge this has been so beautifully deliberated and described by Mike Brown and Joe Goldstein, who initially looked at familial hypercholesterolemia, a disorder just in families, and was able to extrapolate from the pathways involved and the mechanisms involved to a much broader population, essentially everybody with hyperlipidemia. So my question to Rudy is also, as we think about the related disorders and we look at the genetic forms, even of Alzheimer's, and then looking at others, what can we learn from other diseases? And is it possible that a therapy that, for example, raises BDNF, a therapy that does that predictably, would not only be useful for Parkinson's and Huntington's, but potentially Alzheimer's? In other words, something that isn't the cause, but after the cause, the way the body responds is quite conserved, and so the brain too, the response to injury is conserved. Sometimes you want to put a halt on that if you treat early enough. What do you think about the study of less common illnesses, even in this field, and its potential application to more common diseases?
Well, I think you made a great parallel with the amazing discovery of Brown and Goldstein, that they started with a very rare form of cholesterolemia in an early onset family. A gene said cholesterol might matter in heart disease, and people didn't buy it in the beginning. Just like, the first 3 early onset Alzheimer's genes we found in the 80s and 90s, we said it all leads to amyloid as the problem. And it took a long time to get to this point where now we know amyloid does cause the disease, but it does it very early, and the trials we've done were too late. But now we're waiting for the safe drugs. I have a drug that's going into trials next year, gamma secretase modulator, as it's called, that we're hopeful about. There's a company I work with in Boston called AZ Therapies that repurposed a version of the asthma drug, cromolyn, into a format that gets into the brain that's clearing amyloid very well and hitting inflammation. So I think there's hope. I think that the drugs that will be safe, that will get you what you need to do early on, are coming. But if you look across diseases, the greatest commonality, and I have to come back to it again, is that these disease-specific pathologies lead to the death of neurons in small pockets, like Lewy bodies in Parkinson's disease, or amyloid and tangles in different parts of the brain. But the issue is that the brain's innate immune system is primitive. I mean, the brain's immune system doesn't use antibodies and T cells and B cells. It's an innate immune system that's at the same level of an oyster or a horseshoe crab. And when enough neurons start to die in small pockets, the glial cells, glia means glue in Greek, because we used to think they glued the brain together, but they're actually either housekeepers, they're sentinels, keeping track of cell death, and they can be soldiers. And when they see neurons dying, whether it's in Parkinson's, ALS, Huntington's, Alzheimer's, they say, you know what, this part of the neural grid is compromised, and they start shooting out free radicals, they start shooting out cytokines that cause massive astrogliosis and inflammation. And this is where you get the bulk of cell death. And I think the resilient brain studies have really shown us this. When you can see so many plaques and tangles, and these people were lucky enough to have a gene mutation that protected them from neuroinflammation. You know, I do whole genome sequencing of Alzheimer's cases, including resilient cases, and we found mutations in some of the Alzheimer's genes we discovered that control inflammation. We found mutations that were protective, that explained why these people could have so many plaques and tangles and no neuroinflammation. Genes like TREM2, CD33. So I think the common pathway at the end, I like to say this, plaques and tangles in Alzheimer's push you up the mountain. Lewy bodies and Parkinson's push you up the mountain. You can sit there for a good long while, but it's the brain's response to that with inflammation that pushes you off. That's why I'm so excited mostly about we discovered 20 different genes in Alzheimer's that control neuroinflammation. And now those are leading us to drugs like, hopefully the cromolyn or other drugs like it that will stop the neuroinflammation. That's the next big step, I think, in hitting across neurodegenerative diseases and maybe even into psychiatric diseases like depression, where you also have inflammation involved.
Just to add to that, there is a continuum, and I think you've alluded to it.
Yeah.
Dr. Hayden did. This gene C9orf72.
Yeah, good example.
It can produce frontotemporal dementia, language and behavior alterations, or it can produce Lou Gehrig's disease, amyotrophic lateral sclerosis. Frontotemporal dementia can have Parkinsonism associated with it. Alzheimer's disease can have Parkinson's disease associated with it. So this spectrum of neurodegenerative disease crosses a number of established diseases. And I would suggest, rather than emphasize on the clinical manifestations of how a patient clinically is presenting, that we need to go to the genotype. We need to analyze the whole genome of all of these disorders, 1,000 individuals with neurodegenerative disease, Alzheimer's disease specifically, and see what the variations are across the 20,000, 21,000 genes we have. The 3 billion nucleotides in the haplotype genome. And then use that as the strut in terms of what variations occur and how do they correlate with the clinical manifestations. And I think what will emerge is a common set of neurodegenerative phenomena. Neurodegeneration still has to be defined. It is an early manifestation of all of the diseases I've referred to. In my own lab with Doris Lambert-Washington and Min Fu, we were finding in the triple transgenic mouse that there are plaques and there are tangles, but there is also neurodegeneration at the microscopic level. And 10 months later after that occurs, then you start to see plaques. And the same time diversity occurs in individuals, abnormalities in the CSF, as histologic changes in plaques and tangles. And then many years later, plaques occur in a positive scan. So I think we need to do a genomic analysis to define exactly what are the possibilities that correlate with the phenotype and begin to define neurodegeneration primarily, and then how does it differentiate into the named diseases we've referred to.
So I think we're coming to the end of this session. Let me try and summarize what we've learned tonight. I think we've learned about the features of Alzheimer's disease. We've learned about some of the factors that cause Alzheimer's disease and the pathways. We've also learned about how we foster healthy brains and healthy minds, various steps that each one of us can take here. We've learned about the disappointments in some of the trials, but we've also spoken about the hope focusing on neuroinflammation, the hope focusing on inflammatory pathways that actually may talk to a whole spectrum of neurodegenerative diseases. We don't know where the final solution will come, but I can tell you we are where cancer was in the mid-90s or late 90s. we have the biology coming together, and there will be significant advances in the neurodegeneration in our lifetimes, in the next probably decade. And so I see the coming together of genomic science, fundamental neuroscience, together with more detailed description of understanding disease, understanding also of non-medical interventions that preserve brain resilience, and together we can look forward to a much brighter future. And this is the responsibility of all of us, to create societies that are fairer to patients with dementia, to work within our own communities, to do what each one of us can to make that day come a little bit sooner. And I'm going to leave just with one more paragraph from Lisa Genova, because we all know somebody with Alzheimer's or a form of neurodegeneration. And what she says is, being diagnosed with Alzheimer's is like being branded with a scarlet A. This is now who I am. But I want you to know and remember that I'm fundamentally more than that. And this is for all patients, particularly in the early phases of this disease. I'm a wife, a mother, a friend, and soon to be grandmother. I feel still and understand I'm worthy of the love and joy in these relationships. I'm still an active participant in society. My brain no longer works as well, but I use my ears for unconditional listening, my shoulders for crying on, and my arms for hugging others with dementia. That's what we need to do. So I want to thank both Rudy and Roger for sharing their wisdom, their thoughts, their expertise, and thank you all for being here. Thank you to Leslie and to Catherine for allowing us to participate in this special symposium, special opportunity, and I ask you all to thank our colleagues here.
Dr. Rosenberg, Dr. Hayden, and Dr. Tanzi, thank you for joining us tonight. Thank you for giving us hope. Thank you all.